can still. So thanks a lot for joining us. Thanks again. I want to introduce the chair of the steering committee of the Alliance. That's Marie-Christine Wiyad. Marie-Christine, please go ahead. And I'm going to turn off my video, but I'm still going to be here in case anyone needs anything. Thank you.
So thank you all to coming. I'm very happy to welcome you for this webinar. I think it's still time to wish you an happy new year and many success in the implementation of newborn screening in your country. The webinar was requested by some national organizations to support them applying for newborn screening in their country. So the Newborn Screening Alliance advocate to have all babies screened by 2025. It's very important for us to have this baby screen as soon as possible. We try to support all the national organizations, of course, but also all the stakeholders by give them tools to apply for newborn screening. And then I say many thanks to all the speakers that agreed to take time to present and to all the participants. I let the floor to the different speakers and first to Raquel Yayaoui. Raquel, you can present yourself and take the floor.
Hello, everybody, and welcome to this talk, Introduction to SMA Newborn Screening Process and Screening Techniques. My name is Raquel Yayaoui. I'm a medical doctor and clinical biochemistry specialist in Malaga Regional University Hospital, Spain. I would like to thank very much SMA Newborn Screening Alliance for counting on me to participate in this webinar, and I'm very glad to be here. Sorry.
Okay, yeah, your video is off, I think. It was flickering there for a second too.
Can't change it for now.
Okay, no problem. We can hear you fine. Please go ahead, Raquel.
Okay, sorry. Newborn screening is essential public health activity focused on testing every newborn for certain congenital diseases that can result in significant morbidity and mortality without early management. Screening tests are not diagnostic. Rather, they separate newborns at high risk of having a disease from those at low risk, and newborns who have screen positive results indicating higher risk must undergo an additional testing and clinical evaluation to confirm or rule out the suspected disease. Newborn screening programs must be based in ethical principles, guaranteeing equity, informed consent, the protection of confidentiality, and the access to diagnosis, treatment, and follow-up of all infants affected by the screen diseases. Although newborn screening programs have historically focused on screening testing, truly effective newborn screening programs provide an infrastructure for universal access education and rapid follow-up for newborns with a screen positive result. So a complete newborn screening program comprises 6 main components, which are screening, diagnosis, management, follow-up, evaluation, and education. The preferred method for screening used in standard newborn screening programs is capillary blood collected by a heel prick with direct application on a filtered paper. Dried blood spot or DBS specimen is generally collected between 24 to 72 hours after birth. And once collected, the DBS specimens will be transported to the NBS laboratory where there will be carried out the biochemical and molecular screening tests. According to the NBS program protocols, screen positive results should be reported immediately And the newborn screening programs need to arrange or help coordinate follow-up diagnosis testing so the newborns can receive a prompt diagnosis. For a definitive diagnosis confirmation, like for SMA, confirmation genetic studies are usually performed. The aim of treatment will always be to improve the child's survival and prevention of the symptoms in order to modify the natural history of the disease without treatment and ensuring a higher quality of life for the patient and the family. Follow-up will determine whether newborn screening programs have achieved and continue to meet their primary aims of preventing or minimizing morbidity and mortality. And it's vital to evaluating the benefits of newborn screening to an individual throughout his or her lifetime, as well as to the family and society. And newborn screening programs also must establish quality indicators before their implementation begins and must continuously evaluate them in order to identify best practices. Some indicators should be related to the analytical performance of the NBS methodology, like sensitivity, specificity, false positive, and false negative rates, and other aims concerning infants' health outcomes throughout long-term follow-up should be ideally analyzed. An integral component of NBS is ensuring education, awareness and training for all relevant stakeholders. Awareness and education will enable informed participation in newborn screening and will improve parents' experience. And participation in a newborn screening program should be voluntary. The fact that participation in a newborn screening programs is in their child's best interest should be made clear to parents. So in conclusion, newborn screening is not a test. It is a plan for the newborn. It is an integrated health system for secondary prevention of inherited disorders. When a healthcare system evaluates whether newborn screening for a given disease should be made available to the general public, then the severity of the disease, the importance of an early detection that drives a therapeutic intervention with a reasonable risk-benefit profile, and the decision of and the precision of the screening methodology are the main dimensions that will be taken into account. The 10 principles by Wilson and Jagner were published by the World Health Organization in 1968, and they are still commonly used to judge if a disease should be included in a population-based screening panel. Here represent a personal adaptation of these 10 criteria, and it has been shown that SMA meets all the criteria to be included in newborn screening programs, as you may have read in the white paper by the Alliance, where all the criteria are justified one by one. And today we will focus on the screening test for SMA. Newborn DBS specimen for SMA newborn screening can be easily added to standard newborn screening programs without additional specimen collection, and there are no validated biochemical markers of SMA, so DNA testing is the best approach. Several methods based on molecular testing to detect homozygous testing of SMN1 exon 7 deletion have been developed, and SMA due to compound heterozygote mutations that are found in around 5% of individuals with SMA are not identified through the newborn screening process. SMA can be detected from the same DBS punch used to screen newborns with severe combined immunodeficiency or SCID screening, an advantage when it comes to adding SMA to programs that are already screening SCID. And in addition to SCID, SMA can also be combined with screening for X-linked agammaglobulinemia or XLA. Many methods have been validated for SMA newborn screening testing with DBS specimens. They include liquid microbead suspension arrays, high-resolution DNA melting analysis, quantitative real-time polymerase chain reaction or qPCR, competitive oligonucleotide priming PCR, loop-mediated isothermal amplification or LAMP technology, and DNA mass spectrometry. And of these, the technique most used in SMA NBS pilot studies and programs worldwide has been qPCR. The methods for screening can be developed by the laboratory itself, but there are also different commercial reagent kits available. Today we will learn about some of them in the following presentations. Many of these assays for SMA newborn screening are specifically tailored to NBS laboratories, so just modest applications and adaptations and personal training would be required to perform this genetic analysis. Most of these methods are cost efficient, capable of high throughput, and easy to implement in NBS laboratories. Sensitivity and specificity is nearly 100%. So this means that false positives and false negatives are very unlikely to occur, unlike what happens with other diseases that we are already screening for. So these tests for SMA newborn screening have an analytical performance much higher than other screening tests. Each program must establish its diagnostic algorithm based on the first screening results. As an example, I present this one from the state of Ontario. When no copies of SMN1 are detected in the first screening, this result has to be checked in duplicate by repeating the screening test. And subsequently and optionally, a second screening, also called a second-tier test, can be performed on the same DBS specimen to assess the number of copies of SMN2. For this analysis, multiplex ligation-dependent probe amplification, or MLPA technique, or digital droplet PCR are used. According to the newborn screening program protocols, SMA screen-positive results should be reported immediately. For newborns with a screen-positive result for SMA, a rapid referral to a neuropediatrician at an SMA or neuromuscular specialty center for diagnostic confirmation and subsequent information on treatment options is needed. All possible SMA cases identified through newborn screening must be confirmed with a reliable diagnostic test in another blood specimen as soon as possible. And for this case, the MLPA technique is the most frequently used for diagnostic confirmation that should include genetic testing for SMN1, the exon deletions, and SMN2 copy number as a phenotype predictive marker. So thank you very much for your attention.
Thank you very much, Raquel. And now, do you want to introduce Dr. Boomer?
Raquel, can you stop sharing your screen?
Sorry, I'm trying.
Okay, thank you.
Sorry very much. Yeah, of course, I would like to introduce Dr. Boomer. He's a clinical geneticist from the Laboratory of Newborn Screening of Lieja in South Belgium, and he would tell us his experience in the first pilot study of SMA newborn screening in Europe, and he will also share with us his experience in the techniques that they have implemented in their NBS laboratory. Thank you.
Okay, and I think Dr. Boomer might have just dropped out at that point, but I think he's come back in now. So thanks for being patient. Just 1 second.
You need to activate the video, perhaps?
Yeah, there we go. You're a co-host now. Thanks.
Do you see my screen?
Yes, we do.
Yes.
Yes, okay.
In presentation mode. Very nice.
Thank you, Raquel, for your kind introduction. So I am Dr. Böhmer. I am the head of the Newborn Screening Center of the University Hospital of Liège in South Belgium. And I will present you some technical aspects of newborn screening for spinal muscular atrophy. Since we started, we were one of the first newborn screening center to start the screening for SMA in Europe. So I will show you some challenges we face to implement this screening. So what is the objective of newborn screening for SMA? It is to identify homozygote deletion of exon 7 of SMN1 gene, which covers about 95% of SMA patients. So with the screening test, we will miss about 5% of SMA patients. Those patients are patients carrying one exon 7 deletion on one allele and a point mutation on the second allele. So this patient will not be catched by the screening method. So with the screening method, we do not want to identify carriers of the deletion of exon 7 of SMN1 gene. We do not want to identify point mutations of this gene, and we do not want to analyze SMN2 genes. So we only focus on the identification of the homozygote deletion of exon 7 in SMN1 gene. So based on that, we initiated a pilot project which started in our area in southern Belgium in March 2018. And at that time, there were no commercial assay available for screening of SMA. So we developed our homebrew assay to identify these patients. And I show you the technical challenges that have to be considered when you screen for SMA. So you have here, all of you know that for SMA, there are two important genes, which are SMN1 and also a pseudogene, which is SMN2. Both of these genes share more than 99.9% of homology in the genomic sequences. You have here the only two nucleotides of difference in the exon 7 of these two genes. So when you want to identify patients with SMA, you use always a kind of PCR techniques, which is an amplification of the genomic region of interest. And to amplify the SMN1 gene, you will use what we call primers, which are here underlined in yellow. And using the primer, you will amplify both SMN1 and SMN2 gene because the primers are the same for both genes. What is important to identify, it is so SMN1 gene, and you need to use a fluorescent probe, which is here, which will only target and which will be very highly specific to SMN1 gene. And so you will only identify the SMN1 deletion, and you will not identify any aspects of the SMN2 gene. So the specificity of the fluorescent probes is based, in our case, on some chemical modifications of the nucleotides we used, which confers a very high specificity towards the SMN1 gene. So based on that, we initiated a routine implementation of our protocol of the method we have developed. And here is a brief overview of the protocol to screen for SMA. So all dry blood spots from newborns are punched using an automatic puncher into microtiter wells. And then the DNA has to be extracted from the filter paper. So for such to do that, we use a 3-step manual protocol in our cases, which takes about 30 minutes. After DNA extraction, the DNA is transferred into a microtiter plate, which is preloaded with all the reagents that are necessary to run a PCR of the SMN1 gene. So we will amplify the SMN1 gene to make it detectable using specific techniques. And the PCR will take about a bit more, about 1 hour and 30 minutes. And after that, so we can easily transfer the data generated from the instrument into our laboratory information system, into a software to interpret our results. So in total, the overall procedure to screen about 100 samples takes more or less 3 hours. So that's a test we developed about 4 years ago. So here I show you the kind of qPCR results we gather for the 3 types of patients. You have the SMA patients where no SMN1 gene is present, so you don't have any signal correspondent to the SMN1 gene. You have a profile for heterozygote patients where you have you can observe a signal for SMN1 gene, and in green you have the normal patients where you have a bit higher signal than compared to heterozygote patients. And in any case, for all patients, we also use what we call a reference gene, which is important to check whether the overall procedure has worked well. So for all patients, we need to observe a signal for this reference gene, which means that the PCR has worked well. So it's important to validate the results for a patient. So we validated the method we developed using 53 dry blood spots originating from SMA patients, all carrying the homozygote deletion of exon 7 in SMN1 gene. We also used 93 relatives, carriers of the deletion, and 1,000 of newborn samples. And the interpretation of our result is based, in our case, of the ratio of the fluorescence of the SMN1 gene and the reference gene. And you can see here that we calculate a ratio between SMN1 gene and reference gene. And you can see here for normal patients and for heterozygote patients that we have a great overlap between results, which means that we are not able to identify specifically heterozygote patients. That was the aim of the screening. We do not want to identify heterozygote carriers, so that's nice to not identify them. And for SMA patients, you can see that the ratio between SMN1 and reference genes is close to 0. So you have a clear discrimination between homozygote patients and heterozygote and wild-type patients. That's the way we do. Other techniques use a different approach. They don't use a ratio between both genes. They use what we call a CT, which is a threshold above which a fluorescent signal is considered as positive. So that's a different way to interpret results, but both approaches are more or less equivalent. So that's no matter which one you use. How do what I have done? I try to Yes. So here is the flow chart of results we have implemented. When we have a qPCR result which is positive for one patient, we always repeat this result in twice in a second run, and in parallel we initiate from the initial dry blood spot what we call a confirmation, a second tier testing based on MLPA testing to have information on the number of copies of SMN2 gene. And if with both of these techniques we confirm the deletion of exon 7 in SMN1 gene, so we have a positive screening, and we enter then in the diagnostic path. We refer the neonates to a neuromuscular reference center, and where the parents are called back to collect a new independent sample from the neonates and to initiate a confirmatory MLPA testing on this independent sample. So that was a method we developed about 4 years ago. It was a homebrew method. Nowadays, in Europe we face to a new regulation which will enter in vigor within 3 years in 2024, which is the IVDR regulation, which is in vitro diagnostic regulation, which will oblige all clinical laboratories to use commercial methods when available. So we will not be able to anymore to use homebrew methods. So when commercial methods is available, we will be obliged to move to a commercial methods. We already have done that, so we use nowadays already a commercial method, but that will be the case for all screening laboratories who have up to now developed their own methods. They will be obliged to move to a commercial method. And up to date, as Raquel mentioned you in his talk previously, there are several approaches available on the market based either on a qPCR approach, which that I've just shown you. You also have a MELS curve assay, which is also a PCR amplification of your SMN1 gene, but you don't have a fluorescent product. You will analyze what we call a melting curve, which is another way of interpretation of results. You also have a LAMP, which is a loop-mediated isothermal amplification. I think we will have a presentation later on the LAMP approach. You also have techniques based on the mass spectrometry, which are called mass array. So you can also identify the SMA patient based on mass spectrometers. And finally, we also have digital droplet PCR approaches, which are up to now not already available as first-line screening tests, but they are more used as confirmatory testing assays. Then when you have a positive result, you need to confirm it with an alternative method. It's the guidelines. You need to confirm results using alternative methods on an independent sample. The most widely used approach to confirm SMA screening results is the MLPA technique. It's a multiplex ligation probe amplification. So you will identify both SMN1 and SMN2 numbers of copies for all your patients. So with these techniques, you have information of both genes. And you also have nowadays access to digital droplet PCR, which is the alternative methods. And finally, some labs have developed next generation sequencing approaches, which are techniques where you can sequence the whole genomes, but they are focused their approaches to SMN1 and SMN2 genes. So I will briefly present you our results after 4 years of screening in Belgium. SMA screening has now been officialized as regional screening in southern Belgium. So since about 1 year, all newborns in southern Belgium are officially screened for SMA. But during our 3 years pilot study, we screened about 136,000 neonates, and we identified 9 cases with SMN1 gene homozygote deletion. All were treated with different treatments available, nusinersen, Zolgensma, or risdiplam. But we also identified a patient which was not identified by newborn screening, but which was catched on clinical symptoms because he was carrying the deletion of exon 7 of SMN1 gene and a point mutation on the second allele. So that's a patient which could not be caught by the screening, but it was identified based on clinical symptoms. We did not report any false positive, neither false negative during this pilot study, and so we have a prevalence of about 1 to 15,000 neonates with a homozygote deletion, and this prevalence is more or less the same that compared to other initiatives in Europe. So all the patients were treated. We identified 2 patients with 4 copies of SMN2, and we also face a family where the treatment was delayed because due to an initial refusal of the parents to the treatment. And also one family with 4 copies of SMN2, where we could identify the two siblings of 4 and 6 years, which were also carrying 4 copies of SMN2, as they were also SMA type 4 patients. So briefly, newborn screening programs worldwide. So where I will just focus on where are screened patients. So most US in the United States, most newborn so screening is performed by mostly by newborn screening laboratories, while in Europe the screening is often done by genetic labs. It's not directly done by the newborn screening laboratories, but it's the assay is performed by the genetic labs, which is generally the same institutions as the screening lab, but not always. So that might be some add some difficulties in information treatment for patients. So the current situations, as shown you, are the different commercial assays available on the market, and the advantage of the qPCR is that you can multiplex SMA screening with other diseases, like it is the case for SCID screening, but you could imagine to combine SMA screening with screening for hemoglobin disorders. So you can combine it easily with other techniques. But that's the situation nowadays, but in the future, I'm pretty sure that screening will broaden perspectives, and that we will screen more disease, more treatable disease that we can do now using large sequencing techniques, and that's the future we will need to prepare nowadays. Well, I thank you for your attention, and I'm open for any questions. Thank you.
Thank you very much, Francois, for your interesting presentation. So I will remind the audience that they can send us the questions for the presenters through the chat, and we will have a free time for discussing at the end of the webinar. So next, the next presentation will be held by Terry Elenius. She's Global Product Manager from PerkinElmer, Finland, and she will present their solution, new commercial kit for SMA newborn screening. The floor is yours, Terry.
Thank you. I believe you can see my screen now. So
Yes.
Hello everyone, and thanks for this opportunity to speak. So my name is Terhi Helenius, and I work as a global product manager for PerkinElmer's molecular solutions, so including solutions for SMA screening. So PerkinElmer is a global leader in newborn screening, and our reproductive health products have aided in screening totally over 750 million babies. Annually, we screen 50 million babies in over 100 labs all over the world, and every day 70 babies get a better chance to survive their disease that has been detected with our solutions. Our molecular newborn screening tests have been sold since 2014, and next I'll tell you a bit more about that. So in 2014, PerkinElmer was the leader in newborn screening with the first SCID assay in newborn screening called EnLight. EnLight is still the only FDA-approved solution available for screening SCID. And it has screened over 10 million babies so far. So for developing our molecular platform, we listened to our customers and wanted to move forward with the quantitative PCR technology development, and took a step in modernizing our molecular platform in newborn screening solutions to the qPCR platform. And at the same time, the awareness of SMA started to gain interest in the newborn screening laboratories. So as the clinical investigations show a direct correlation between early diagnosis and a better prognosis of the disease, the newborn screening programs, or many newborn screening programs, have been initiated in several countries. So therefore we heard this need from our customers, and our solution for them was the qPCR solution called Ionis. So we provide an option with Ionis for SMA screening with this platform. The assay is a multiplexed assay, so it means that it can also screen for SCID and XLA from the same DBS bunch if needed. It is a total CE IVD solution, meaning that it includes all instruments, reagents, and tools for the analysis and the reporting of the results. The key features of this product is that it's configurable and flexible, which means that it can be accommodated to meet the needs of different laboratory sizes by just choosing the appropriate instrumentation setup. Ionis is available both in 96 well and 384 well formats, and both manually and automated workflows are validated and available. The solution also comes with a dedicated software for the analysis and the reporting of the results. And most importantly, this CE IVD solution is based on a robust qPCR core chemistry and has already been used for screening more than 1 million babies successfully. So with our current molecular portfolio, we are addressing the newborn screening need for SMA and also SCID and XLA globally. So for example, in Australia, more than 250,000 babies have already been screened for SMA during the past 3 years with our solution. And in Europe, our products are used in several countries as SMA screening is emerging at the moment. So as I mentioned, one customer for SMA screening is from Australia, and they have been working with us from the beginning of their implementation of SMA screening. And so far, more than 250,000 babies have been screened for SMA. The rerun rate has been confirmed to be less than 1% here. And so far, 22 SMA screen positive samples have been identified, and the median age at diagnosis has been 15 days. The median age of treatment start has been 25 days. And this study also indicates that the SMA incidence in Australia is approximately 1 in 10,000 screened babies. Then I'd like to go back to talk about how PerkinElmer has responded to the newborn screening needs over the years. So we have now quite a lot of experience from using qPCR in newborn screening, and we still know our customers and their needs very well. So therefore, the next step here was to improve the overall experience of the qPCR screening workflow. So what is coming next is a simple qPCR platform that will be accessible to all kinds of labs. This new platform is called EonisQ, and initially it will include a more simple screening solution for SCID, XLA, and SMA screening. So how did we end up with this next product in our portfolio? So we again listened to our customers and built the new EonisQ platform based on customer feedback. So these were the main building blocks to consider for being able to deliver a product that would serve the needs of newborn screening labs. So the most important building block was to ensure a robust core chemistry of the product. And we are basing the chemistry of the new EonisQ platform on the current Eonis product, which has, as I mentioned, a track record of 1 million successfully screened samples. The next important building block was to create a product with a short turnaround time. And we wanted to make this product accessible to all, regardless of the size of the lab, the PCR experience of the personnel, or the actual physical areas available in the lab. So from there, the idea was to create a product that was easy to implement and easy to train. This means that we want to support a workflow that does not need heavy instrumentation, that does not require long installation processes and ongoing services. So also we did not want to limit the customer lab spaces to the need of a designated PCR clean room. We also want to provide our customers a product that is easy to store and ship. And yes, so all of these points were very important for us during the development phase, because we of course understand our part and responsibility in keeping the continuous screening ongoing. So therefore, the new EonisQ product is based on dry qPCR technology, where all the qPCR reagents are pre-dispensed and dried on the PCR plate. So in short, the system includes a short DNA elution step with no washes, a sample to PCR plate pipetting step, and then the actual PCR cycling step. The needed instruments are simple and robust PerkinElmer instruments, and they will be validated for a full CE IVD solution. The dedicated software will be the last building block here, ensuring that the product is easy to use by everyone. So all in all, the final result of the development work based on direct customer needs is what we are soon launching, a product that has a unique and simple workflow without compromising the core chemistry. The turnaround time for the whole assay is less than 2 hours, and the reagents support working in normal laboratory spaces without specifically PCR trained personnel. And when will this be available? There will soon be a research use version available of the kit, and eventually a CE IVD solution as well. Thank you for your attention, and feel free to ask any questions or just contact me later on if you have something you want to discuss. Thank you.
Thank you very much, Terry, for your presentation. Next, I would like to present Liselot Determerman. She is an R&D manager from LaCAR MDx Technologies, which is a company from Belgium, and she will share with us their solution for SMA newborn screening. Thank you.
Hi everyone, I hope you can see my screen. So I'm Liselot from the LaCAR company, and I'm responsible for the scientific department in the company. And so I'm going to explain you a little bit about the SMA screening test we developed in the company. So maybe just to start, I will present a little bit the company LaCAR, because it's maybe unknown for most of you. So we are a Belgian company, which was created in the end of 2013. And the goal was to be one of the first companies to offer an easy solution for genetic molecular routine diagnostics, because there were a lot of assays on the market, but most of them need are expensive, or they need a lot of equipment and a lot of investments from the lab to get the assay up and running. So the idea was to develop assays for SNP detection, so single point mutations without DNA extraction, and all of this using the LAMP technology. So this is an overview of the company until today. So as I said, we started in the end of 2013. And in 2015, we got the R&D department up and running, and we had the development of our first kit without DNA extraction for a single point mutation in thrombosis. In 2016, we continued our developments, and we marked our first kit CE IVD. And then afterwards, we also get the ISO 13485, which means that we can produce also all products on site, which is done until now. In 2017, so the goal was to broaden the range of products we could offer to our clients. And so we developed kits for pharmacogenetics, food intolerances like lactose, or autoimmune diseases. And then in 2018, as the panel of products we had to offer became larger and larger, we tried to get more and more routine diagnostic labs to work with our products. And our initial focus was Belgium, France, the Netherlands, and Switzerland. In 2019, people start talking about screening for SMA. And when looking at the screening of SMA, we thought that it could fit the LaCAR products. And so we started the development of our SMA kit. And so today, beginning of 2022, we have our CE IVD mark for our SMA kits. We have a large panel of kits to detect other human genetic diseases. And we have more than 100 routine diagnostics lab working with our assays in all of Europe. What we have planned for 2022 is to promote SMA screening of newborns by performing a large-scale SMA study. So we are sponsoring a pilot study in 10 different sites, and in total, we will carry out 200,000 tests just to demonstrate the feasibility of routine use. So how does it work theoretically, the assay of LaCAR? So it's a screening kit for SMA without DNA extraction directly from the dried blood spot. And there's an automated interpretation at the end of the testing. All the necessary reagents are provided within the kits that we sell. So there's a lysis buffer, a reaction buffer with all the components you need to perform the reaction, and is ready to use. And of course, a control to make sure that the reaction went well. So as I said, for our kits and also for the SMA kit, we use the LAMP technology. The LAMP technology is similar to a PCR technology. It has the same goal to amplify a specific region of the DNA. But the LAMP technology has several advantages to qPCR, that it's really simple, it's robust, it's cost effective, and it has a high speed. One of the differences with qPCR is also that there are 6 primers needed for amplification and not 2. So this adds to the reaction specificity. And at the end, there's a high amount of DNA that is produced. So for the SMA kit, using the LAMP principle, there are primers present to amplify specifically the exon 7 around the exon 7 of SMN1 to detect the deletion of exon 7. There are primers amplifying an internal control or a reference gene, just to make sure that the reaction went well. And then we added also a sequence-specific probe to detect a point mutation which is present in exon 7 and which causes the conversion of SMN1 to SMN2. So after amplification, we need to have a detection. The detection is done by a melting curve analysis. And so we have 2 options. Or you have the detection of SMN1, which means that SMN1 exon 7 was present. Or you have an amplification of the internal control and no amplification of exon 7 of SMN1, which means that exon 7 is absent. And so you're SMA positive. After the analysis of the amplicon, we can also take a look at the presence of the point mutation within exon 7. And so here we will make the difference between the presence of the point mutation or the wild type. If there's a presence of a point mutation, you can also be SMA positive. Now, how does this work in practice? So this is our workflow, which is quite simple. So we start off with a regular 3.2 millimeter punch in a deep well plate, and we add just 400 microliter of lysis buffer, which is present in the kits. We let it stand 1 hour at room temperature. After this hour, we mix and we transfer 5 microliter of the lysis sample to 20 microliter of the ready-to-use reaction buffer. We give a quick spin just to avoid all bubbles, and we launch in the qPCR machine. We have 2 platforms that are CE IVD, the Quant Studio and the Light Cycler. In total, the run on the qPCR cycle takes around 1 hour. So this means in 2 hours, you can get a 96 well plate from sample to results. And then each hour you can get an additional plate, results from an additional plate. As I said, there's also an automatic interpretation using a software, which is called the GeneFox software. So the software will import the raw data file from the qPCR cycler and will give you an automated interpretation. It will combine, which I explained, the 2 types of detection, amplicon detection and point mutation detection. So no need to go look in detail to all the graphs on the qPCR machine. The automated interpretation will be done in the GeneFox software. And so you will get a result for SMA negative or SMA positive. And there will also be an indication if the run is valid, invalid, based on the controls that were present in the run. The GeneFox software also gives you a full traceability with sample names, which scans the barcode on the plate. So everything is fully traceable. And of course, there's the possibility to export a report in PDF or CSV which can be also connected to the system, the laboratory system. So as a conclusion, the test of LaCAR can detect the exon 7 deletion of SMN1 and the conversion due to the point mutation. We do not detect any carriers, which is important, as Dr. Boomer explained. The assay works directly from the dry blood spot. We do not do any DNA extraction, only a simple lysis step. Due to this simple workflow, there's only limited pipetting steps. And this means that there's a limited risk for any manipulation error, and also a limited risk for contamination. Also, as the workflow is really easy, there's a possibility to automate it on any pipetting robot. And a big advantage is also the automated interpretation using the GeneFox software. As highlighted also in other presentations, is that the exon 7 deletion is not the only cause of SMA, and so other mutations can be present, and like the other methods in our SMA kit, they will not be detected. There's also a risk of false positives. As with every method, something can go wrong during the pipetting or during the run. As there are controls present in the kits, normally there's an identification of invalid results. But if any false positives would arise, we recommend, as is quite normal, to repeat all positive results from a new dry blood spot. And then, of course, as this is a screening assay, there is also a confirmation afterwards. For the moment, LaCAR has only an SMA kit on the market, which is CE IVD. However, we have a SCID assay that is in development, and that should join the SMA kit in our newborn screening panel, and which should be compatible with the SMA. So as a final conclusion, LaCAR has a CE IVD solution on the market. And we have an international study for SMA up and going, where 10 sites will be doing testing for newborn screening. 200,000 tests will be included, and we will show that the newborn screening for SMA is feasible, and that this is also feasible using the LaCAR method. So thank you for your attention, and if you have any questions, you can ask me afterwards. Thank you.
Thank you very much, Liselot, for your nice presentation. We're looking forward to seeing that results from the SMA international study. And now I would like to present the last speech by Frida Olive. She's head of sales and marketing from Immuno IVD, which is a company from Sweden, and she will talk about their solution on SMA and SCID newborn screening kit. Thank you. We can't hear you at the moment.
Sorry, now I should be unmuted. Can you see my screen as well on my presentation?
Yes, it's fine.
Perfect, thank you very much. So I'm Frida Olive, and I'm very happy to tell you a bit more about Immuno IVD's solution for SMA screening. Let's see if I can change screen. So Immuno IVD is a Swedish company. We're located just outside of Stockholm. The company was founded in 2015, and 2 years after that, we launched our first CE IVD product. We are specialized in qPCR-based assays, and we are specialized in newborn screening. So that is what we are 100% focusing on. We're also targeting the European market, so we are currently providing our assays to several countries within Europe. Our screening assay goes under the brand name Spot It, and they are available in 3 different versions. We have the spotted TREC screening kit that is screening for SCID. We have the spotted TREC and CREC screening kit, which is screening for SCID as well as XLA. And finally, we have the latest addition to our spotted assay, which is the spotted TREC and SMN1 assay, which is a combined screening assay for SCID and SMA. And this is, of course, the product, the reason why I'm here and presenting today. All the spotted assays work in the same way. From a laboratory or handling perspective, there is no difference between the different kits. So in short, it takes about 3 hours to go from a Guthrie card or from a dry blood spot to having the screening results. Our assays are based on real-time PCR, which makes the assay very sensitive. And also it is a very transparent data collection, meaning that everyone who has some experience in qPCR can easily understand what is happening. The main focus when we have been developing our assay has been on the part that comes before the qPCR step. So we have spent a lot of time on making a laboratory procedure that has little hands-on time, because that's very critical in a screening laboratory. So you don't require so much time from the laboratory technician, but at the same time, we have wanted a product that enables the lab to work without investing in an expensive automated handling equipment, so a pipetting robot. So the way that we have been able to do this is that we are providing in the kit plates that are pre-filled with reagents, which means that there is no need for the laboratory themselves to either prepare the reagents or to dispense the reagents into the plates. We also work with a very special way of transferring reagents from one location to the other. And we do that by using this plate stacking method, as we call it. And that is what you can see on the picture to the right here. We have multiple plates that are stacked onto each other. And by using centrifugation, we can transfer liquid from one plate to the other. And I will explain that in more details shortly. So we've had this several times during the meeting today, but what we use as a marker for SMA is the exon 7 in the SMN1 gene. And in about 95% of all SMA patients, there is a homozygous deletion of this exon. We will only detect homozygous deletions, which means that we will not detect carriers, which is, of course, is of high importance in a screening assay. So some of the users that are using the spotted assays are shown on this slide. We have, for example, Karolinska University Hospital, the laboratory in Sweden that are analyzing all the newborns in Sweden. They are currently using our spotted TREC assay, meaning that they are screening for SCID. In Switzerland, all the babies are screened for SCID as well as XLA. In the Netherlands, all the Dutch babies are currently screened for SCID, but they will soon also be screened for SMA. Charité in Berlin, in Germany, is using our combined SCID SMA assay since of October 1st, when SMA screening was implemented in Germany. So this is just an example of some of the users. So if we look a little bit more into details in the laboratory procedure of the spotted assay, it can be divided into 5 different steps. The first one being the sample distribution, which is very similar for all the assays that you've been hearing about today. Here we take the Guthrie card or the dried blood spot or the filtered card where the newborn's blood has been collected into spots. We take a 3.2 millimeter punch out of this and we add it to the filter plate that comes with the kit. So almost all of the plastics that are required for this workflow comes with the kit. So one of them being this filter plate. The filter plate is a plate with 96 holes in it, and in the middle of each hole you have a filter, which is shown on the zoom-in picture here to the right. On each plate, you can analyze 82 newborns, so 82 blood spots can be punched into this filter plate. The remaining wells are used for controls and calibration curves. So after punching, you will have this small little punch where the newborn's blood is available, and in the newborn's blood we have the DNA, which is what we will analyze. The next step is a washing step where we want to get rid of hemoglobin and some impurities in the blood. This is done by adding 200 microliters of a rinse solution that comes with the kit into each well. So that's a fairly large volume that is easy for the people in the lab to pipette. You can do it with a so-called multi-pipette where you're adding the liquid to multiple wells at the same time. We have the solution, we let it be for 20 minutes, and then to remove the liquid, we don't pipette, but we put this plate into a centrifuge, and the liquid will go through the filter, leaving the DBS or the punch at the top. We will repeat this once again, but this time we will only leave it in room temperature, and it will only be for five minutes. We add the liquid, we spin it in a centrifuge, and what will be left is a clean DBS, or clean punch. When that is done, we want to get the DNA out of the punch. And to do that, we will take an elution plate that comes with the kit and that is pre-filled with an elution solution. We will take this one, we will flip it on top of our filter plates, and in between we will use this frame, and the frame are there to keep both plates in a tight position, and also to make sure that the plates are put in the right direction. We take this plate bundle and we will flop it. Then it will look like we see here. On top, we will have the filter plate with a clean punch, and at the bottom, the elution plate with this elution solution. We give it a quick spin in the centrifuge, and the little punch will fall into the elution solution. We take that plate, put it in 95 degrees for 30 minutes, and that will cause the DNA that's previously in the punch will migrate out to the elution solution. Next step is to prepare for the qPCR. Then we will use another plate pre-filled with the required reagents. So the qPCR plate comes with a master mix pre-filled in it. It contains enzyme, it contains primers, probes, nucleotides, and other things that are required for the qPCR reaction. We will then transfer the eluted DNA, which is in the elution plate. We put the elution plate on top. We have a filter plate in the middle, and at the bottom we have the qPCR plate. And in between plates, we have these frames to ensure the position and the orientation of the plates are correct. It will look like this, like you see in this picture here. We will spin it in a centrifuge, and the blue solution here that contains the DNA will go down through the filters and down into the PCR reaction. And with that, we have a solution we have a plate that is ready to go into the qPCR machine where the qPCR can be performed. So I hope this showed how straightforward it is and how you can work with a large number of samples without the need for either manual pipetting or therefore the need for an automated pipetting system. So when it is done, you put the instrument, as I said, in the PCR machine. It will take about 1 hour before you have the results. And the results will look something like this. We will have three colors of curves, where the red one is indicating that the baby does not have SCID. It's a marker, the trait that we are looking at. The green line here is our reference gene that we use to ensure that everything has worked as it should. We use a reference gene called actin beta, or ACTB. And the yellow lines that you can see is indicating that there is the exon 7 of the SMN1 gene is present. If the SMN or exon 7 of the SMN1 gene is not present, it will look something like this, indicating that there is no or very little amplification. So when you have this result, the first thing to do is a quality control or quality check to ensure that everything has worked. And if it looks okay, you can verify the results from each newborn. Different labs do it differently. Some labs are using this kind of data or exported a version of that, adding it into their LIMS system where the analysis and the interpretation is happening. Sorry, the interpretation of the analysis, But we also have a software that is currently available as a research use only tool, but soon it will also have the CE-IVD mark. And if you use this software, there will be it's very easy to interpret the data. The software will automatically look at the entire run and say, okay, everything looks fine. We see all the controls are showing up as they should. And then it will look at each of the individual samples. If the sample is negative for both SCID and SMA, it will show you a green light here. If it's positive for either of the disease, there will be a red dot, and then you just click on that and you will see if it is SMA or SCID that the baby is positive for. And if something has not worked with a reference gene, you will get a yellow signal, and that can be an indication that there has been some technical errors or the sample hasn't been enough DNA in the spot, for example. If you have either a red or a yellow spot, you should rerun that sample. You can take the same filter plane, same Guthrie card, take 2 new punches and redo the analysis. And if those one is still showing that there is no amplification of SMN1, it is the screening is positive and the baby should be referred for further investigation, investigating the SMN2 gene numbers or number of SMN2 genes, for example. That was a brief introduction to the assay that Immuno IVD is providing. My contact details are here, but for question, I think I will hand over to the organizer and to Raquel. Thank you.
Thank you very much, Frida. Now I think it's time to make a short summary from the presentations, and please send your questions to the presenters through the chat. I would like to remark from all the presentations, thank at first all the speakers for sharing their experience. And now I think in conclusion that we can say that NBS process for implementing SMA will need to design a screening and diagnosis algorithm, and also the program will need to develop a clinical and therapeutic protocol. So remember that newborn screening is not only a test, it's a plan for the newborn. We have seen that there are different methods for SMA newborn screening. And please remember that these screening methods are designed only to detect homozygous SMA patients for the deletion of exon 7 of SMN1 gene. And these methods will not detect other type of SMA and also will not detect carriers. We have seen that there are many commercial solutions. And one of the advantages is that we can add SMA to programs that are already screening for SCID. We have seen that the companies that offers diagnostic products for SMA newborn screening have made a high effort that these solutions are now very easy to train and easy to implement. And they also present a very high analytical performance. They are very sensitive and specific. Some of them that we commonly use are based on qPCR or LAMP technology, as we have seen, and they may present no DNA extraction need or DNA extraction without washes. So the idea is that could be implemented easily. They also have developed a generation of interpretation software with these methods that facilitates the work of NBS laboratories. And don't forget that we will need a confirmation when we get a screen positive result, and this confirmation should be performed in an independent blood sample, probably by MLPA technique or the digital droplet PCR. Some laboratories use a second screening in DBS to assess the SMN2 copy number, which will help to predict phenotype. And we have already some clinical validation results, as Dr. Francois Bommer presenting in their study, pilot study from Belgium. So in conclusion, I think that these methods are suitable for newborn screening. And I would like to thank everybody, because I think that the companies of the diagnostic services and all the stakeholders should work together. And we have made a significant step to continue advancing in the implementation of SMA newborn screening in Europe. So thank you very much. And now it's time for questions.
Raquel, we had one question from Marie-Christine. I don't know if she's still here and she can ask herself. Yeah, there she is. Good. Marie-Christine, I think it was for Terry about blood tests, right?
Yes, I have a question for all the company, more or less, because we are facing a very important number of tests to do, and I would like to do how many tests they can do by day or something with only one machine. Do we have to buy 10 machines to have a large population cover, or only one machine can cover? How many people can we cover, or can we do a test with one machine by day?
Yeah, maybe I can start by answering. So with our new EONIS Q system, it's for 96 samples per run, and one run is less than 2 hours. And you can of course start the elution of the next round while you're doing the PCR of the first run. So I would say that you would be able to do at least 5 rounds per day. So 5 times 90-ish samples. And with the current EONIS solution, you can do either 96 or 384 samples per run. So, and this one can be automated as well. So if you have a very big lab, then you can process many samples during one day.
Okay, thank you. Is it more or less the same for the other technique?
On the Immuno IVD side, I would say that with one equipment, you can run between 4 to 5, depends a little bit on how you put up or how you plan the day. But probably like 4 runs, I would say, could easily be managed with one set of equipments, and that times 82 to get the number of samples. So just below 400.
Okay, thank you.
For our test, it should be a little bit more. It takes 1 hour to do the lysis and 1 hour on the PCR platform. So each hour you can launch a new run. So it could be around 6-7 plates a day.
And each run is around?
It's a 96 well plate with 2 controls, so it's 94 samples.
Okay, thank you very much.
I don't see any other questions in the chat. I know this is a pretty complex, or I find it as a pretty complex subject, but I thought all the presenters really did, and Raquel, especially with her summary, everyone did a really good job of making this pretty understandable. And we hope that this can be a basis for further learning. So yeah, we'll be having this recording available for you, as well as the slides of some or hopefully all of the presenters after this. And yeah, just once again, thanks for joining and taking part, and thanks to all of you for presenting and sharing your expertise with the SMA community tonight. It's very much appreciated.
And just before to conclude, just I want to show you something I have received today by the post, because there's still no possibility of meeting, but I have received this trophy for the SMA Newborn Screening Alliance, and thanks to all the company who are there, because it's with the support of the company, and also Raquel, thank you very much for your support reading, preparing the paper, the white paper. So thanks to everybody, and thank you very much. And I'm very proud to show you this trophy.
Congratulations.
So I hope everybody is now well informed about the different type of technique they can implement in the country. If you have any question, no problem, you send us the question when you see another time, and we try to answer to all the questions. Send your question to Robert, and we will answer. Thank you very much to everybody, and I say good evening. Bye.
Thank you.
Bye-bye. Thank you. Bye-bye.
Everyone.
Thank you. Bye.
Bye.
Ciao.